Good morning. I'm Joan Slaughter, the founder and executive director of the Morgan Adams Foundation, and thank you all very, very much for joining us this morning for our first ever virtual What's Up Doc. We appreciate you joining us in this forum as we give it a shot and see how it works. We realize we're coming together in this format because of our concerted effort to shelter in place and be safer at home as we work together to keep our community safe during these unprecedented times. So thank you very much for being with us today in this way. Silver lining is that, of course, normally I would be thanking you for getting up very early in the morning to join us for What's Up Doc, and I'm happy to say by starting in a virtual forum at 9:15, we all got to sleep in a little earlier, so I appreciate that. Speaking of the coronavirus, I realize that COVID concerns are very much on everyone's mind, and while we're not specifically addressing that during today's forum, we will be able to share with you after our session a webinar hosted yesterday by our friends at Solving Kids Cancer and the Max Cure Fund, where they spoke directly to the concerns that cancer families may be having regarding their children's treatment and care during this crisis, as well as the impact that COVID-19 is having on clinical research. So we'll send you that link and we'll post that video up on our social media channels. Thank you. For this morning, our mirror agenda is our typical What's Up Doc agenda, which is really framed around an objective to share with you more insights into the work that your support is funding and how laboratory research is driven both by the questions our docs are seeing in the clinic and then how the laboratory work that they're doing can then also be translated into outcomes and improved overall treatment and care for children battling cancer. A little bit of background, excuse me, before we get started. I know we have some new people joining us. So the Morgan Adams Foundation was actually started after about 21 years ago, after my oldest daughter, Morgan, was diagnosed at the age of 5 years old with a particularly aggressive brain tumor. And this was in 1997, and at the time there was very little treatment advances being made for pediatric brain tumors. And so after she passed in 98, we decided as a family that we wanted to get more involved in what we could do to help the doctors treat children with cancer in a better format overall. We decided that funding research, which was very limited at the time, was the area of our particular interest. And so we started partnering with the research team at Children's Hospital Colorado and figuring out how to build the basic biology infrastructure that would support the work in developing new treatments for kids with brain tumors. Over the course of the last 20 years and our partnership through Children's Colorado, we have also been able to grow a network across the country through different research collaboratives and be able to really start to have an understanding about how we can improve overall research for children across the country and around the world. It is an honor to be able to work alongside these positions, some of whom I have the privilege to introduce you to today and to share the work that they're doing and the very powerful and amazing dedication they bring to helping fund better outcomes for kids with cancer. Before we get started on the science side, though, we like to make sure that our community is aware that our work extends beyond just Morgan's story. We have also had the distinct honor of meeting hundreds of families who are going through the same journey that my family went through. And every year when we do What's Up Doc, we ask one of our ambassador families to come and share with you the story of their child's journey and the impact that cancer has had on them. So this morning we have with us Heather and Dave Mahnken, tomato, who are here with us to tell you a little bit more about what it was like for Eli as he was diagnosed with cancer and as he went through his treatment. Heather and Dave, thank you both so much for being with us this morning.
Thank you, Joan. Good morning. My name is David Mahnken, and this is my wife, Heather. And this morning, we'd like to share with you a bit about our son, Eli, and his journey. In March of 2017, our 21-month-old son, Eli, stopped using his left arm. After initial x-rays of the arm, nothing was found, and we were told it would likely resolve on its own. April 18th, Eli was unable to turn his head, and we took Eli to Children's Hospital's ER. where they found a mass in his brainstem extending through his neck. During the next week, we took family pictures. We got Eli his first haircut in preparation for the upcoming surgery, not knowing his outcome. A week later, Eli underwent a 10-hour surgery to remove the tumor. They were able to remove 90% of the tumor. After the surgery, Eli was unable to lift his head, had lost control of the left side of his body, and was able to sit up or to walk. After 2 weeks of intense therapy, Eli was learning to walk again, and we were feeling hopeful. May 5th, pathology came back that Eli had a juvenile pielocystic astrocytoma, a rare but benign brain tumor not likely to regrow or require further intervention. We were so relieved by the news. 2 weeks later, genetic typing of the tumor came back, and it had some very interesting mutations not seen in kids. They weren't certain what that meant for Eli's prognosis at that time. A month later, during a follow-up MRI, we had devastating news that the tumor had regrown over 50%, and we needed to start radiation immediately to buy us some time to come up with a plan of care. Eli had 28 treatments of radiation to his brainstem and neck over the next 6 weeks, requiring Eli to be sedated each day for that procedure. Post-radiation, Eli began taking a new drug called trametinib that they weren't even sure would work due to specific mutations, but there really weren't any good options for us. The good news was trametinib worked. It stopped the tumor's growth. So for the next 2 years, we learned to manage the side effects of medication and radiation. We stopped taking the drug in September of 2019 to give Eli his body a chance to heal, because they just didn't know what that does to a kid long term. Eli continues to have MRIs every 3 months, and we continue to have positive news with stable scan, which means no growth or changes. We're truly thankful for whatever time we have with Eli, and we look forward to each day as a blessing.
Being told that your child has a brain tumor is devastating. However, there's nothing more gut-wrenching than being told that the tumor is back, that the tumor's unique and rare mutations has made it very aggressive, and they don't know how to treat it. And your little boy, who just celebrated his 2nd birthday, is in a grave situation. What do you do as a parent? I felt paralyzed, I felt helpless, and to be honest, I lost hope of what the future would be like for Eli. Let's be real. How do you find hope when the cards that you've been dealt seem so bleak? And the next question, how on earth do you make the right choice about moving forward? The medical team shared with us that they strongly felt that Eli would not respond to traditional chemotherapy, and the side effects would be limiting and decrease his quality of life. The discussion next about how to move forward were terms that were not in my lexicon. Terms like emergent radiation, compassionate use, photon radiation, proton radiation, experimental, off-label, were all thrown around, and my head was spinning. But what happened next was pretty remarkable. Dr. Nick Foreman, Eli's primary neuro-oncologist, brought in a simple white piece of paper with chicken scratch written on it. It was an innovative treatment plan based on the work from his research lab. He proposed that we try a targeted oral chemotherapy drug, a MEK inhibitor called trametinib. Trametinib was originally developed for adult melanoma and was starting to be used with children with brain tumors. It was at that moment I had my first glimpse of hope. After starting the medication, Eli continued to make gains. And for our family, we were given the most precious and priceless gift, time. Time to make memories, time to enjoy those special developmental milestones, time to just find joy in living. You see, I call that little pill that Eli took every day a miracle drug. We didn't think Eli would make it to see his 3rd birthday, but he did. And today, he's a thriving, vivacious, talkative, and happy 4-year-old, almost 5-year-old little boy. And as a family, we've spent the last 3 years creating memories of traveling, camping, and hiking. The most memorable memory I have of Eli right now is teaching him and watching him learn how to downhill ski, which he continues to improve each season. For our family, research has given us the gift of time. And if it was not for the efforts and the generosity and the mission of the Morgan Adams Foundation, our story would have been something different. We are so grateful for the foundation and their work to make childhood cancer research possible. You have made a tremendous difference in our family. Thank you.
Thank you very much.
Thank you both so much for sharing. I appreciate the opportunity to have you tell Eli's story. So thank you very much for that. I got to see the video of him skiing downhill the first time, and I have to tell you, when that text came through, it was very rewarding to know that he was able to have that experience. So thank you. It's always incredibly emotional for me to listen to other people share what it's like for their kids to go through this. I relive a lot of Morgan's journey when I listen to our families speak. So I get a little bit to cough a little bit and clear my throat, but thank you very much. And Heather referenced a miracle drug, and I think the thing that's really important for us to share with our donors is that the miracle drugs don't come miraculously. They come with a lot of work and a lot of effort. Part of the technology that the foundation has been funding it relates to deep sequencing of different cancer cells to help inform what the different mutations and fusions might be that are creating the cancer. In addition to that, there's some screening technology that has been put into place. We want to show you a little bit more about the science that happens and what actually goes into making a miracle drug. To give us that information, I'm going to pass the screen over to Dr. Nathan Dahl. Nathan is a recently appointed assistant professor who has done a lot of clinical work in neuro-oncology, but is establishing his laboratory because he sees the importance of research and how it informs clinical outcome. So without further ado, Nathan.
Well, thank you for that introduction, Joan. And thank you so much to Heather and Dave for being willing to share the story of Eli's journey. Like Joan mentioned, I am similar to most of our investigators, both a physician within our clinical neuro-oncology program, as well as a laboratory-based researcher. And I feel like it's important to highlight that from the very beginning, because it means that each one of us knows firsthand how awful it is to have those conversations, like Heather just described, with a family to describe the limitations of the treatments that we have available. And that really informs and motivates the research that we do, which is largely focused on trying to find new ways to treat these tumors. It is really the incredible track record and infrastructure in place here at the Morgan Adams Foundation and University of Colorado program that brought me here and led me to set up my research lab here, because I think it's an incredible place to not only do that sequencing and do that research and do that discovery, but then to move that into the clinic and treat patients, which at the end of the day is what we all care about. But before I talk a little bit about how that process works and how we go about that research, I want to give you a sense of what we're up against. So the most recent classification schema that came out identified more than 50 different types of brain tumors. That's 50 different kinds of diagnoses, 50 different biologies. And that's before incorporating the very specific, very personalized mutations like the Mankins described. There are, as of this morning, more than 150 FDA-approved anti-cancer drugs, with several times that in the preclinical pipeline. And if you do the math, what that means is that if we just want to test 2 drug combinations, that's already in excess of 1 million possible permutations. And when you realize that many of our successful treatments involve 4, 5, 6 drug regimens, that very quickly becomes prohibitive. And the reality is that drug companies aren't interested in doing that testing. The National Cancer Institute really isn't interested in funding that testing. And it comes down to academic organizations like ours in partnership with philanthropic groups like the Morgan Adams Foundation to actually push those things forward. So to give us a sense of how we are leveraging some of our new technologies and new techniques to tackle those astronomical odds, I want to talk today about really two different tools and complementary techniques that we're using to approach that. These are both approaches that we describe as high throughput screenings, which means that rather than testing a specific drug against a specific tumor a million times over, these are techniques that let us test thousands and thousands of different genes, different drugs, different combinations, all simultaneously. And there are sort of two pipelines that we use both independently and in combination to help answer those questions. On the left here is a schema of what we refer to as genetic screening, where we use advanced genetic techniques like RNA interference or CRISPR gene editing to target thousands and thousands of genes at a single time. And then through very convoluted sequencing approaches, let us find new targets that kill cancer and kill tumor cells that we can then pursue further for drug treatments. And on the right is a parallel approach, which is high throughput drug screening, where we use machines like this multiplate reader here that was actually purchased for a group through the Morgan Adams Foundation that lets us test hundreds and hundreds of drug combinations and doses and dosing schedules at a single time in a way that would have previously taken us days and days and days to even set up those experiments. And we can use those techniques in parallel to try and overcome what really otherwise would have been some insurmountable combinations. So I briefly am going to just talk about three different projects that have been born out of that, that are moving through the research pipeline and making their way out of our lab group and actually into the clinic. So the first here is research done with medulloblastoma. This is the most common aggressive cancer that we see in children, in brain tumors. And this is a genetics-based approach, that first pipeline, where we were able to do deep sequencing of dozens of patient samples that were donated by families, just like the Mankins, towards our research. We identify thousands of transcripts from those tumors and then combine that with a high throughput screening approach to identify a target called WEE1. And WEE1 is a gene that helps regulate how those tumor cells divide, and found that when we target that with drugs that are already available, that we can kill those tumors. That's shown effectiveness in preclinical testing and is currently going to be opening up in clinical trial in children. The second project here came out of the Foreman lab, Eli's neuro-oncologist, and this is the other half of that pipeline, so the high throughput drug screening approach, where we were able to take hundreds of those FDA-approved clinical drugs and screen them against a dozen different tumor models, and then enrich those for combinations that were specifically effective against this tumor of interest, this ependymoma. And that identified a combination of drugs, 5-FU and retinoic acid, that are already available. They are already FDA approved and on the market, show effectiveness against a tumor that has proven very difficult to treat. With following the additional testing done in the Foreman lab, this combination is now also being moved forward as a proposal for a clinical trial. And then most recently, this is some of my own work that's combined the two of those approaches in a tumor type called diffuse intrinsic pontine gliomas. These are some of the worst tumors that we see in that they are categorically incurable. They're fatal for every child that hits them. And as of right now, we don't have effective medical therapies. And what we did is we used that first approach of genetic screening to target thousands of genes that have potential for implications in that tumor. We identify a list of targets from that screening approach that we think are relevant. And then we combine that with a drug screening approach against those targets to find new agents that are effective against the tumor. And in doing so, we found a drug Indicated here, this AZD4573 that targets a gene called CDK9. And in preclinical models of DIPG has been very effective in treating tumors and would be transformative if we were able to bear that out in clinical trials. This is currently under conversations with AstraZeneca, who's interested in moving this forward into children as well. And what that means is that Children's Colorado doesn't look at just what we can do here locally, but we look at nationally and internationally. We are part of these phase 1 consortiums, which are groups of children's hospitals across the country who take care of children with brain cancer. And as of our last meeting this fall, we had 4 different trials that have come from discoveries within our lab group and are now being moved forward into the clinic and into patients. This pipeline coming through the phase 1 consortium is where the drug that treated Eli started and came through this program. And our labs are continuing to work on the next generation of these proposals so that we have more and more miracle drugs like that for the kids who are still waiting to get them.
It's really kind of fascinating to have that information distilled down for us in a way that makes it possible for me to understand, which is always a bit of a challenge. So I appreciate that, and I appreciate you walking us through the background on how new drugs are investigated against very specific cancers. So thank you very much for that. That process is starting to emerge in some popular writings. We hear a lot more references to personalized medicine and targeted therapies. So joining us next is Dr. Leah Gore. She's the section chief for the Center for Cancer and Blood Disorders at Children's Hospital Colorado and the newly appointed vice chair for the Children's Oncology Group. She has a background in developing novel treatments for pediatric malignancies, and so she's uniquely qualified to talk to us a little bit more about personalized medicine, what that means in this day and age, new drug development, how these things are being brought forward at Children's Colorado and actually across the country and internationally as she serves on a multitude of committees. So thank you very much for being with us, Dr. Gore. I'll pass it over to you.
Thanks. Good morning. It is a pleasure and a privilege to be part of this. I am very much looking forward to sharing just a small bit of information with you. I'd like to start, first of all, by really describing what it means to try to move personalized medicine forward for every child with cancer. And when Joan had originally asked me to be part of this, I told her I couldn't be here because I was due to testify in front of Congress about the importance of funding genomic testing for children with cancer and having insurance companies be obligated to pay for this. And I think Eli's story really embodies the importance of being able to have this information because many tumors look very similar under a microscope. But many tumors, as you just saw by Dr. Dahl's really lovely presentation, do not look very similar the deeper we dig. And the deeper we dig, the more individualized almost every tumor looks. There's certainly different groups of tumors that look similar, but very much these have individual, what I would describe as molecular fingerprints. And I'm going to share just a very brief slide to try to demonstrate that even further from what Nathan just talked about. This picture here, I'm hoping it's projecting okay, is essentially a genomic fingerprint, a molecular fingerprint of a particular tumor. And if you look at the depth of what each of these lines means and the layers going both up and down and forward and back, this is essentially a 3-dimensional scan of what one particular tumor looks like. And if you look at the detail in this, it really, I think, describes why we need to understand what each of these things, each of these bars, each of these colors might represent. Because until we understand those things, we may not know the exact, precise, best way to treat. And that's why this is called precision medicine. So I think it's just a really good example of why we do that. I'm going to go back just to the basic screen here to really talk about why shouldn't we do this? This seems like such an important, critical piece of information to have to assess every child with cancer. And so why shouldn't we do that? And I thought I would highlight just a few of the barriers to that. Dr. Dahl already described some of them. Some of them are very practical, some of them are much more theoretical. But our goal at Children's Colorado and many other institutions that we collaborate with around the country and around the world is to make this sort of technology available for every child so that every child gets an absolutely personalized roadmap to be able to treat their individual cancer. When we do that, we have to think about a number of things. One is financial. As I just mentioned, can we get an insurance company to pay for this testing? The testing itself can range anywhere from $500 to $10,000. Many insurance companies don't pay for this. And so that's a very simple barrier. From the research perspective, what are the practical kind of operational perspectives? We need to have somebody in the operating room, as Eli's mom and dad talked about. That was a 10-hour surgery. We need to have somebody waiting essentially in the operating room to take that tumor sample and get it immediately to the lab so that it is the most precise, exact replica of what is going on in a child's body that we can then study in the lab. There are ethical concerns around this. There are things that we might detect on that genetic fingerprinting that might reveal other chances for developing certain diseases. So this testing isn't specific necessarily to a cancer cell, but it can reveal other diseases and other problems that we may not even understand how to deal with at this time. So we need a full team of bioinformatics experts to interpret the data with us. We need a full team of genetic counselors to interpret the data with us and to understand what are the implications of these test results so that we can provide a family and a patient the best and most accurate information. This takes a team of hundreds, literally. And so not every institution is able to support that degree of faculty, of staff, of volume, of 24/7 coverage to be able to achieve this. But we think that that's the right way to do this. We think that touching all of these touch points, touching each of these elements is the best way to make discoveries that are going to eventually help us not to have to tell a family like Dave and Heather that their child has had something really terrible happen. We think that that's the only right way to do it, and we'll continue to think that that's the only right way to do it. And it is through the efforts of people like Dr. Dahl and Dr. Foreman and all of the team that they are part of and all of the team at Children's Colorado that helps make this happen every day for patients. I had the privilege of taking care of Morgan. Joan and I have known each other a long time. And I have made a promise that I will not stop working at this until we figure out how to do it better. So I think that this is really important. And to distract just a little bit from this message, I think that the importance of what the Morgan Adams Foundation does in moments like this, when we are crowded by COVID and paralyzed by research slowdowns because of having to contain lab personnel, when we have volume issues in the hospital, when every hospital in the entire country and in the entire world is facing dramatic financial challenges, the only way to make up for this is with philanthropic effort. And so I was joking with Joan earlier today, there has never been a time when the Morgan Adams Foundation efforts and work has been more important. It is a commitment that we all have. Nathan can tell you, he's been working at weird hours from home, and we've been working remotely for the last 6 weeks to try to contain this virus. But I have also told people that COVID does not pay attention to cancer, and our patients do not pay attention to everything else that they need in the middle of all of this crisis nationally and internationally. So the work of the Morgan Adams Foundation and the kind of work that you've just seen in the story of Eli and all of the wonderful news that has come from that do not take a break because of this unprecedented health crisis. The financial impacts of COVID right now have never been more significant to research and to our ability to take care of patients. So it's a brief detour from what molecular and personalized medicine is. And at some point I will have the opportunity to stand in front of Congress and talk about why this is important. But for today, I hope that that message is carried forward to you. I really appreciate the time that you've taken to be able to participate in this, and I am happy to always be available to this group for additional information or things that we can do to help underscore the importance of what this is. So thank you.
Oh, thank you, Leah, very much. It's not nice that you mentioned Morgan in that context and how long we've known each other. It's hard for me. It's amazing that 22 years later, I get as emotional about it as I do, but it doesn't go away. So Leah mentioned the work of the Morgan Adams Foundation, and I will tell you that we have over the last 22 years had the incredible, amazing opportunity to build a phenomenal community of supporters who have come forward to make the work that we do to support our lab partners possible. To talk to you a little bit about what it takes to be part of that community, we're bringing forward the chairman of the Race Against Kids Cancer, which is an event that we partner with the Rocky Mountain Vintage Racers on. Kirk Peterson has been involved with that event for the last 8 of its 10 years and has willingly taken on chairing it, which means he has to work with me a lot. So he's a brave soul, and he's here to tell you a little bit more about why he is involved with Morgan Adams Foundation and a little bit about the race. So Kirk, thanks for being with us.
Well, as Joan just said, I'm involved with a racing club that's had a relationship with the Morgan Adams Foundation. This is now our 10th year. And over those last 9 years, we've raised over $1.2 million. And we try to do in the last few years over $200,000 a year. And what I'd like to talk about is ways that all of us can participate in giving. And there's the 3 T's of contribution: time, talent, and treasure. And I'd like to start off with the time. Time is one of those things that we cannot make any more of, but we all have plenty to give. And I would just encourage you to think about ways that you might be able to help as a volunteer. Get in touch with the Morgan Adams Foundation, sign up to do that. They have a number of events throughout the year that take quite a bit of man-hour to plan and execute. And it's just a wonderful way to be involved, meet these families, meet these kids, and really contribute something of value. On the talent side, we all possess some expertise. Sometimes that's very unique. There are professional services that many of us could donate, depending on what it is we do for a living. There are things we know, experiences we have that can help contribute in a way that is unique. I remember one time when our racing club was invited to Children's Hospital, where we were going to be involved with a demonstration of some of the equipment that our particular club's fundraising had funded. And listening to the doctor show us this piece of equipment, explain everything to us, after about 15 or 20 seconds, it was all over my head. I had no idea what they were talking about, but that's okay. Not all of us are called to be doctors or researchers, but we do things and know things and have particular talent that we can contribute as well. And then the last one of these, of course, is treasure. And this is where we give money. And I found that it's extremely easy to solicit funds for this. No amount is too small. I have a little old lady client of mine. Every year she gives $20 and then tells me, I hope you win the race. She's just fantastic. But it doesn't really matter whether it's $20 or $20,000. Any amount of money is fine. You can ask all your friends. You can ask colleagues, co-workers, clients. I don't pre-qualify anybody. I've determined that nobody is going to think less of me, nor would they of you, if you ask them to help out in finding a cure for pediatric cancer. And if somebody does think less of you, you just don't want that person in your email address book anymore. As far as the other methods, social media is a good way to get the word out. People can give one time, they can give recurring. And then there's also things that are less thought of, like planned giving. It could be part of your estate plan. People that have to do required minimum distributions out of their RIAs can give qualified charitable deductions for that. So there's a lot of creative ways that you can give. But I've found that there's some interesting things that happen when you get involved with the Morgan Adams Foundation. Sometimes we think, oh, this is a great cause, I can help out with this, and then something unexpected happens. And what that unexpected thing is that you find out it enriches your life as well through this involvement. I've met some fantastic families. I've met some great people at the foundation. I've met some wonderful little kids. And I have to tell you that that has made an impact on my life for the last 8 years I've been involved with this. And every single day that 43 more kids are diagnosed with cancer just punctuates how much research is needed, funding for that research is needed, in ways that each and every one of us can help. So I encourage everybody to think about how you can give, how you can help, and how we can help take care of finding a cure for pediatric cancer and doing it through this research. Thanks very much.
We wanted to thank you very, very much for joining us this morning. I think we as a team were very focused on trying to make sure we kept this broadcast effective and efficient, that we were able to relay for you a lot of information in a short period of time, but to also be conscious and cogent about the work that you're supporting and the impact that you are able to have by partnering with us as we push forward more and more research looking for better outcomes and hopefully more miracle treatments for kids like Eli. So I think we're concluded for this morning. We would continue to ask for your support as we move through the rest of this year, as we all together as a community determine how we can best effectively raise the financial funds that we need, the funds that we need, the time and the talent to be able to move forward the incredibly important research that our kids need. So we would ask very much that you consider how you can get involved with pushing the work forward, and come and join us when we're able to gather again. We look forward to seeing you all in person. And in the interim, we will continue to provide you updates on the research that you enable through all of our social media channels, through our website, through webinars like this, and through sharing communications from our sister organizations who are also in the fight alongside us. So Heather and Dave, thank you. Dr. Gore, Dr. Dahl, thank you all very much for being with us to share your work and your stories and your thoughts and your passion with our greater community. And to all of you who joined us this morning, the last I heard there were 115 people out there joined with us. Thank you for being here. Thank you for your comments. Thank you for your unwavering support of our work and the support of our families and our community. We hope you have a beautiful day. If you're here in Colorado, it's supposed to be gorgeous. So please get outside and stretch your legs. And we look forward to seeing you online and in person very soon. Thank you very much. Take good care. Bye-bye.